So hello everyone, my name is Daofeng Li. So I'm from Team 1's lab at WashU. So I'm very glad today for the opportunity to show our work at this 14th scientific webinar series. So I will talk about the genomics visualization as a title on the WashU Epigenome browser. When people are talking about genome browsers, I think the first thing on their mind is probably the UCSC genome browser. So WashU Epigenome browser is a large genome browser software. We started this development since 2010. So it aims to be a lightweight and UI-friendly genome browser with the latest web technology. So because it's lightweighted, if you want to install a WashU browser mirror, you just need to have this JavaScript environment installed, then it can be just installed in less than 5 minutes. So, and this is very large the user interface for our current browser, and also this a URL for the browser. So on the top, it has a menu which you can control the regions and tracks and have functions. You can settings, and also you can have links to the help documentations and source code as well. So after that, you have a menu. After that is like you have a menu. You can allow you to jump to different genomic locations. Then on the middle part is like the display of different type of tracks in different display mode. Like you have heat maps, you have wiggle style, you have color bar, line plot, etc. So along with this, there's like also some of tools and color metadata map is provided as well for you to like configure your visualization. So, but like with the development of current, so many new biotechnology, so many new assay types. So we are thinking the browser function need to be expanded, not just display 1D annotation and then 2D view data like high-C data. So I think that we are thinking the genome browser functions should be able to say, for example, can show the 3D genomics data, right? So you can, the browser and the 3D view, they can be interactive with each other. And you should be able to view image data and show data dynamics with the time, which is our 4D support. It can also function as a platform for like new collaborations, new function development, right? So after this environment, in the next, I will talk about all the features focused on the 3D browser. And in terms of 3D, in terms of new data types and new more functions, I will show you how to visualize the long-read methylation data on the browser and some recent development of a new tool called Comparative Genomics browser. So the first thing I'll talk about is about the 3D browser, and here's the design principles for the 3D browser. So for the 3D coordinate, since the 3D coordinate model provides like new coordinates for the genomic data, so we designed a file format called G3D data model, which we'll talk about it more in the next slides. So basically, a 3D model is like with XYZ coordinates is in a binary index format. So, and then the 3D model and the genomic data are decoupled, which means the 3D model just represents one single cell state at a single time. So we can display multi-models together in the browser. So all the functions in the 3D browser should be also available in all the functions on the 1D browser should be also available in the 3D browser. Like in the 3D browser, you should be able to zoom in, zoom out, and you can search and query, right? You can label and select different region to display. You should be able to show the genes, expressions, or the regular analysis result like big wig format, common states in the 3D browser as well. You should have like some collection between this 1D and the 3D views. So some of the functions are only possible in the 3D browser. You can show compartments, text annotation, loop annotations, right? So we are also continue develop this 3D model to make this dynamic from 1D to dynamic track. So like I mentioned, we designed like G3D file format to host the 3D model, which is like an indexed binary format for faster retrieval of 3D data over the web. So So this we also developed this command line tool called the G3 tools, and this can be available in the Python package index, and it can use to convert any 3D model with XYZ coordinates to the binary G3D format. So G3D tools can pass like bed-like files like output from other software like Pastis from Biolobos lab, and also the nuclear 3D format from Gemma's lab. And also the 3D output from DeepSeek HiC tool. So this format supports like remote range requests, hosts many metadata, different resolutions, and haplotypes, cells, and samples, etc. And also we have like Python and JavaScript API provided for you to generate this file and read the file and get data out from the file. So like so next slide is like an animation shows you the 3D view overview, how this 3D view looks like in the WashU Epigenome browser. So when you load the G3D file to the browser, so you can do basic operations like including zoom in, zoom out, rotate, use your mouse, and then you can control the model display. Like you can toggle the display of different haplotypes. You can say you can only show paternal 3D or only show the maternal 3D model. So by default, the 3D view is highlighting the current browser region when you are loading, right? So you can disable the feature, or you can change a different highlight region or style. You can also manually like choose a gene symbol or genomic region to highlight. So other than that, we can use like numerical or annotation data to paint the 3D structure. So in the video, we are using CPG density to color the 3D structure with the color gradients. So the colors can also be configured. You can say choose a different color for high value and low value, but also those colors can be configured. We can use gene annotation, cytoband, com-HM state, HiC loop, or domain annotation to paint the 3D structure as well. And in this video, we're showing examples showing we can use chromosome AB compartment data from 4DN and also com-HM states annotation to paint the 3D structure. You can see these segments are painted in different colors based on the annotation we provided, either like AB compartment annotation or like com-HM annotation. So in this example, I'm showing you how you can use the 3D browser to view loops and domains, which is kind of specific for the 3D view. So I just want to, if you can see my cursor, you can see there's like in the A part, top part, there's like a typical browser view with the ruler indicate the genomic locations. Then there's like a compartment annotation, which is like AB compartment, but you also have a subtype A1, A2 from B1 to B4 compartments. Then you have a domain annotation, which is showing a different color, indicate different domain. Then there's like a HiC data here, which is, you can see there's like a loop indicating this rectangle here. And then if you go to the same 3D structure and the same part of the loop, which is enlarged here in the left part, right part, so you can also see the corresponding so this 3D structure, both from maternal corresponding to this HiC loop. And also the anchor points, which is showed here, the blue, is displayed here in the 3D model as blue spheres. So basically, this blue sphere indicates the loop anchors in the 3D representation. And if you show in a linear browser, and this is the two small ticks shows the loop domain boundary. And also the color here shows you purple, white, and green. And it shows here purple, white, green here in this 3D model, how you can like fold this 3D structure based on the HiC data prediction. So same as well. So basically, if you use this compartment data, you can basically to paint this whole 3D model either in this particular region, which is here, and also either in the whole chromosome. You can see the colors 3D structure are colored use the compartment annotation. So in the bottom is pretty much similar idea, but we are switching the domain annotation. So you can see this like same genome browser view with ruler track and a domain annotation with each color bar indicate a different domain annotation. And then there's a HiC map if you zoom into this particular region, this high-C map is like domain here, and then particularly the 3D structure, the whole chromosome structure, you can also zoom in to get fish out this particular 3D structure for this particular region. And also in this L example, I'm showing just an example showing the nearby domain annotation for this MYC gene here. So the 3D browser view was used to like recapitulate several findings from published papers. So in this panel A, we can see a haplotype resolved high-C maps are shown in the linear regular linear browser, which is like the top we have maternal data and the bottom we have paternal high-C map. So this like orange line indicates this super loop anchors, right? And then there's some green dot indicates the super loop anchors only available in the paternal sample, but not in the paternal sample. If we use these two data to like modeling 3D structure and display the 3D structure together, you can see this is the result 3D structure from maternal and paternal. And then we are using red spheres to like indicate the superloop anchors. So basically, this example shows you since the paternal sample we have more superloop anchors, and this result a more dense 3D structure from the 3D model. Other than the paternal, it's like more loose because it has much less superloop anchors in this high-C data as well. So this shows you different, basically, high-C structure are pretty different out of 3D model. In the right part, we're showing the browser view around a mouse source line gene locus, and this typical ruler gene density, and also this test annotation, and also this shows you three high-C distance metrics from 3 cells. So and then predict 3D structure. So the 3D structure also colored based on the test annotation provided in this paper as well. And also this top, this right bottom shows you basically the same idea, shows you like two different high-C maps from two different cell types, and also the different high-C maps and different 3D structures can be pretty out from different high-C maps. And this is showing the data from chromosome X region. So yeah, sorry. Okay. So yeah, this sort of summarize these 3D functions. So I'm just quickly go to mention, like I mentioned in the first slides, or besides display 3D structure, the browser can also display the image data. So like we're showing how you can show the image from the IDR in the browser. So because some of the image in the IDR, they have like they have genes or like probe locations associated with each image. So that's how we make this connection from image to genomic coordinates. So when you view a genomic region, so the images associated with this region will be displayed in this grid style. So you can click the image, will give you the metadata like the plane name, well name, and channel information also in the tooltip. And then there's also a link to the OMR server. You can click it, which will bring you more information, shows you the image. But this is hosted in the IDR website. So we are also supporting some of the 4D images. So you can load the 4D image hub in the browser. You can also see from the screenshot. We have like 4D data, 4D image data loaded in the browser as well. So in order to support the fourth dimension of data, so we made this data animate, so it's like a movie. So in the first movie, the high-C movie, where we are showing the 8 high-C contact maps as a function of cell cycle, and visualize them as an animation. So data is coming from this paper in JobDex group. So this is showing 8 high-C maps in the region in chromosome 14. So in the left, in the top below left, there's like some labels showing you different time points for different high-C maps. So basically it shows you how the high-C maps, how this genome organization was changed along the time. So not only the high-C data can be animated, so annotation and also numeric data can be displayed in the animated way, like in the right bottom movie, we're showing a dynamic numerical track, showing the signals animated from 3 cell classes of some single cell data. So with all these components, 3D structure, image data, or displayed in the browser, we split the browser UI into a multi-panel settings. So the browser user interface display data from different types in each panel, right? Representing in different dimensionalities. So the panel on the left is showing the main conventional browser window. The panel on the right shows you the 3D view and shows you a 3D genome model. And the panel on the bottom right shows you an image from a microscope experiment. So in this we're actually showing like a dynamic track, but this screenshot, it cannot show animation here. It's kind of like an animated version for these 3 top tracks here. So with these settings, we're showing all the data from 1D to 4D in one single web page. So now I'm going to jump to my second topic, which is about the visualization of comparative genomics. So here's some background information. It's like comparative genomics is about based on the alignment of linear genomes, right? You align the coordinates and compare the sequence. You can, based on the alignment, you can compare the genetic and similarity and difference between between the species you aligned. You can also check the base pair level substitution and the large scale rearrangement events based on the alignment. So this work called like Comparative Epigenome browser was led by WashU in our lab. Is trying to compare data from multiple species on the genome. So you can select 2 or more species and no data mapped to each species, and it will display all of the data collected by Sentinel. So this shows you an example creating a cattle mouse comparative browser view and use it to compare DNA methylation in heart, lung, and liver tissues between cow and mouse. This work is recently published in Genome Research. So I'm showing you how we actually, for the genome alignment track, how the visualization is done. So this example shows you between human and mouse, AGZ8 and MM10 alignment, how this alignment is displayed at different zooming levels. So on the top, it's kind of like a base level, right? If display this human and mouse, AGZ8 and MM10 alignment at the nucleotide level. So sequence, those the strand information in alignment, it displays these arrows. These arrows indicate the strand forwards or might reverse strand. And then the synthetic nucleotides from these 2 species are vertically aligned. And also there's like gap inserted. So if there's like a match, so you can see like this vertical lines ticks indicate there's a match, but it's a mismatch. There's no line, no ticks here. So if you sort of zoom out, continue to zoom out a little bit. So when the computer spend 1 pixel doesn't enough to represent 1 base pair in the screen, we're going to show the sort of a similar settings, but we are not going to show the base ATCG basis anymore. But when you mouse over, you can see the alignment. If you mouse over the region, you can see the alignment in the nearby few bases. If you continue to zoom out, so like that means like we don't have enough space to render like more detailed information. So we're going to just use like blocks to represent the synthetic blocks from mouse and human. We'll just connect it use this Bezier curve to display on the browser. So with this alignment settings, it's actually much easier to like show the 3D genome structure difference between species. So in this example, showing the human lymphoblast high-C contact map, which map to AGZ8, and also gorilla lymphoblast high-C data map to the GOR4, will compared by anchoring to the human and the gorilla alignment track. So you can see this sort of like synthetic regions, and you can compare the 3D structure difference between these 2 species based on the alignment. So using this function, we are very glad to collaborate with RenLab to hosting their portal for supporting comparative epigenome in the mammary brain, which supports the brain data from 4 species. And this website can be accessed from here. So I'm quickly go through my last topic here. So my last topic was about like long read methylation visualization. So before just like a little bit background information. So it's like overall, like cytosine methylation is like an important epigenomic mark. So in mammals, most of the cytosine methylation occurs in the CpG context, right? So CpG methylation is like a cell-specific regressive marker, so it plays an important role in gene regulation. So we all know, like right now, most bisulfite data were based on the bisulfite sequence, but right now the latest non-read technology also can give you the methylation data without doing any bisulfite treatment. Right? So for example, using the Oxford Lalapro, you can call CpG methylation using ONT reads. You can measure current changes, do not distinguish not only the ATCG basis, and you can also difference the C and the methyl C. Same thing for no same thing, but it's just like PacBio also can do this. Like PacBio CpG methylation calling, they are using reaction kinetics to infer methylated CpG site. Okay, so all these long-read methylation data somehow, but it's like right now they are stored in sort of a BIOM file. And then methylation calling data are stored in this MM and ML tags of BIOM files. So people right now, most people are using IGV to visualize this long-read methylation data in IGV. And this is an example screenshot for the IGV view of long-read methylation data, and shows you the promoter region of the gene, and shows you the low methylation, which is indicated in blue color for low methylation and the red color for high methylation. So, I mean, IGV view is good, but it's just sometimes it's hard to deal with the huge size of BIOM file. For example, typically the BIOM file size is about 500 gigabit. So with that, so we are sort of displaying a like lightweight file format called modbed format, and also the associated with modbed tools. So which just capture the necessary information from the BIOM file, makes the file size much smaller. So this file format just contains 8 columns. You have like a chromosome start, end, position, and the rename and ID. Something you can tag back this read. To this optional score, you can use it to like for sorting. You have strand information. Then the last 2 columns is the most important. You have methylated basis position and unmethylated basis positions. This is the data for the methylation data. So with this file format, we are like display a new track type called a modbed track. You can so this kind of like at a base pair resolution, we display the non-read methylation using sort of this cycle dot-like visualization. So in each line here is represent a read, and the field and the empty circles represent methylated and unmethylated bases. And then the orange and the blue color indicate the different strand, information, forward strand or a reverse strand. So if you zoom out, when you just like lose enough space to represent one base CpG site in this computer screen, we're going to summarize a bunch of CpG methylation to a bar level. So in this read level, we're also showing the read methylation based on each read level and show you summarize based on multiple nearby CpG sites. If you continue to zoom out, so we're going to continue to summarize from different reads as well. So the basically different, if we go different view region, we're going to summarize different type of data to show in order for effective visualization. So in the read level, we also like support IGV style view. You can configure the style to be looking like IGV, exactly like blue for low methylation and red for high methylation. Yeah, this pretty much sort of summarize my visualization. I just want to mention we have been collaborating, a lot of collaboration happening in the past few years where people, our browser users developing plugins, right? So, so users from our community, they display new track types. So say, for example, the one is published in 2021 called QBAD, and for the visualization of point person data, and another one published last year called Dynamic Second Track for showing context-specific features for each nucleotide. So so I think I hope the function for the browser I showed you today will be useful in your research sometimes in the future. So, and this is all I have today. So I want to thank all the consortium we will and are involved, including 4DN for the funding sources. We are in Twitter, so welcome to contact us for any questions or feedbacks. So in this background, well, I'm showing you some of the 5-6 data showing visualizing the board tract. So thank you again, and I'm happy to take any questions.